All right, welcome everybody to something new that we're trying on CNBC Cures. This is a way for us to really reach out and talk about the stories that matter to our community. And today we are starting with a story that matters very much to me. It's very near and dear to my heart. Camp4 Therapeutics has been working on a therapy to treat Syngap. Australian regulators have cleared them to begin phase 1/2 trials for a new ASO therapy that they're working on for patients with Syngap1. Syngap is the disease that my daughter Kaylee has. If you've been watching on Cures, you know a little bit about her and what we've been so interested in. Josh Mendel-Brem is the CEO of Camp4 Therapeutics, and I'm going to start asking you questions from the perspective of a parent who cares an awful lot about the stuff that you're working on today. Thank you for what you're doing. This is pretty incredible news because we've never had a therapy to treat Syngap1, and this is the first time that we're going to see human trials toward that end. Why did you get involved with Syngap to begin with, Josh?
Yep. Before I dive into that, just real quickly, Becky, thank you for having me and Camp4. And, I'd be remiss if I didn't actually say we probably wouldn't be having this conversation if it wasn't for the parents, the patient community, the thought leaders, scientists, physicians that actually not only got the word out, but actually have furthered the science, done natural history studies, done things that really pave the path for these things to happen. So it's actually more the companies thanking the parents and all the work that's been done there that is allowing this to happen. So it's a good collaboration, and it's the way things hopefully should work, to be able to let industry do their job and have opportunities to create therapies for these patients that need it. So
Josh and I, by the way, Josh and I have never spoken before. This is the first time we've actually communicated in any way, shape, or form. But obviously, I've been watching what you've been doing for a long time. This was a huge cause for celebration in the community.
Yeah.
What happened with this regulatory permission that was granted by Australia?
Yeah. So we. Couple comments. You said it's a phase 1/2 trial, so maybe just to put a little more information around that. So our intention is to take our drug and go directly into patients, because we think there's an opportunity for a benefit there. In addition, we've been very thoughtful about how we go about doing this. So we've taken a global point of view. There's patients everywhere with this disease, and the things that we considered are where are the sites that have expertise in delivering the drug? It's an intrathecal delivery, meaning it goes through the spine. Where are the thought leaders? Where are their strong patient communities? And where is there regulatory precedent for using antisense oligonucleotides, or ASOs? Australia has a few different features going for it. First and foremost, it has one of the leading experts in Syngap1, Dr. Ingrid Scheffer, who was actually one of the first to describe the disease clinically. It also has a very strong patient community. And as importantly, Australia has set a really nice precedent as being able to have a streamlined process to begin treating as quickly as possible. So it is the first in a set of dominoes to essentially take a global approach to our phase 1/2. And I'll just put a plug in and say we've also gotten approval to start in Argentina as well. So That's another one of the jurisdictions, and you'll hear more about others as the year goes on.
I mean, you talked about some things that are really important, to have the experts who are there, to have the patient population. And then I think what's probably driving all of this is the regulatory setup that allows for these things. I mean, I look in the United States, there's a big patient population, and there's a huge expert population here as well. I guess it's a little tougher to get through the U.S. FDA at this point?
Well, look, the FDA's and the U.S. are a critical part of our strategy, right? And it's really just a matter of timing, that's all. And so, as I mentioned, we've taken a global approach. The U.S. is part of this. We started with the ex-U.S. jurisdictions, which tend to just allow things to be a little more streamlined and move more quickly on the front end. So that includes Europe, Australia, Argentina, as I mentioned. The U.S. just takes, and all the jurisdictions take a slightly different approach, to be fair. The U.S. just requires a bit more to get started. So as an example, ex-U.S. says we only require one species of tox, for example, monkeys. The U.S., for example, can say we'd like monkeys as well as mice. So things like that, and just in terms of how they like to be engaged. So what I can tell you is the U.S. is absolutely on our radar. We are
Okay.
planning discussions with them, and hopefully we'll have a few sites in the U.S. as well, following what we're doing now.
The toxicity studies that, again, shows you if these things can be safely handled by patients that come through. Yep. I thought you had 2 different toxicity studies, one for mice and in non-human primates. Am I wrong on that?
You're completely correct. It's just that what I was saying is ex-US jurisdictions actually don't require that.
Okay.
They are comfortable with just saying if you show us it's safe in monkeys, that is perfectly fine for us. The U.S. tends to take a more robust approach and says we'd also like to see it in mice, for example, or another species.
Is the thought that you start these phase 1/2 trials in these ex-U.S. jurisdictions and see how that goes, and then use that to kind of lobby the U.S. FDA on what you would be doing here?
It's a simple question, a complicated answer. I think based on our data to date and the precedents that have been set, we feel really good about approaching the FDA in terms of being able to start. So yeah. I think on one hand, it certainly helps that the ex-US jurisdictions are giving us the green light to start, and we'll have the opportunity to share what those details look like. But in and of itself, I think we feel really good about being able to approach the FDA based on, one of the advantages of using this chemistry, this ASO chemistry, is there's been hundreds of clinical trials with it. There are approved products on the market with this chemistry. So we get to benefit from that. And it helps us create a really good plan to work with the FDA and make sure we can have as fast a path as possible.
All right, let's talk through the trials themselves. And I don't know how much information you can give us. I haven't been able to find anywhere how many patients you plan to enroll in this, how old those patients will be. Is that something you can talk about?
Yeah, I can talk about it, and I'll give a clue to when we'll share more information. So right now, our intention is to treat at least 30 Syngap1 patients in our phase 1/2 trial. We have flexibility to potentially do more than that. Mm-hmm. And again, we want to take patients from multiple different jurisdictions, not just Australia. Look, this is a disease, as you know well, where the earlier you catch it, the better. That's true of many rare genetic diseases. It just matters. But yeah, we believe we could do more than just Syngap, but we want to get this right for Syngap. And our intention is to get as young as possible with our treatment here. And so there's always a balancing act of safety as a first priority here to make sure, we create a safe drug. So we're working with regulatory agencies to design the study in such a way where maybe we start, if you will, at an age that's, 8 to 12 years old, for example, for the first patients. And then we drop down as quickly as possible to get to younger patients. And so right now, if you were to eventually look it up, it would say ages 2 to 18. But, our strategy is to try and get to patients as young as possible to start with.
Right. Just for people who aren't really versed in how this stuff works, the ASO therapy, as you mentioned, it's an intrathecal injection, meaning it's basically a spinal tap that you have to get.
That's right.
But is it every 3 or 4 months that has to be re-injected?
Yeah, so just 2 comments there. So that's correct. Essentially, one of the benefits of the chemistry is it lasts in your system for a long time. Mm-hmm. So typically, based on drugs that have already been approved, you get it once a month for about 3 or 4 months, and then you can spread it out once every 3 to 6 months. Every drug is a little bit different. What I can also put a plug in for, which is really good actually, is the first drug that really took advantage of this approach was Spinraza for spinal muscle atrophy, Ionis and Biogen. They really did a wonderful thing for the industry and for families. And now it's become a much more common way of delivering the drug, and we're benefiting from that as well, and in terms of its safety and robustness as far as physicians and sites being able to administer it.
Right, because it's really difficult to get at a lot of these diseases because you've got to cross the blood-brain barrier.
That's right.
Things that have to happen. The science of this is really pretty fascinating. You all are the first ones who are using this regulatory RNA, the strategy of going after the regulatory RNA to try and increase the production of the protein from the good gene. Most of the ASOs that have been trying to work on this have just gone after Syngap itself. This is a step removed. Instead of going after the Syngap, you're going after the regulatory RNA. Explain what the theory is for that, why, because is it easier if you're going after regulatory RNA, or is this something where you feel like, okay, if we get this to work, it can be a platform that can work on a lot of other diseases as well?
Yeah, it's a wonderful question, somewhat related to what you had asked in the beginning, which is, how do we find our way here? And so Our technology acts at the gene level, meaning essentially we can turn the faucet on more for the healthy remaining gene. That's really helpful as well because Syngap in and of itself makes different versions of the protein. So by acting at the gene level, we essentially hold constant the different versions of the protein that can be increased. So we think that offers a benefit because you're essentially putting back into the system what nature already designed it to do. So that's the advantage of our approach. We think that, to your question, it can be used across multiple different types of diseases where you have one remaining healthy gene, and that gene needs to essentially be asked to put more protein back in the system. And, we started with Syngap for a few different reasons. One is the science has been very well understood. Credit, again, to the academics and the patient community for pushing that out. The unmet need really spoke to us. There's a lot of patients with this disease and they deserve better, and there's nothing out there for them. And we felt it was a really nice application for our technology in that it could have a transformative effect for these patients.
How many patients, how big is the patient population as you see it? Because this is a pretty small patient population. I know the last numbers I saw were 1,700 people around the world, but we also think it's pretty underdiagnosed.
I think you're completely right. Maybe anecdotally, I can just put a plug in. I've spent 20 years in rare diseases. I started at Genzyme. There are rare diseases that you'll hear about. You may never meet a patient with that disease, okay? And that's very, very rare. What I can tell you about Syngap is I've met many of the patients, and there's been many patients that have shown up through colleagues and, my wife's friend has a daughter or son with this disease. So yes, it's rare, but my view is it's not as rare as people think. We've guided to say we believe there's at least 10,000, if not more, patients in the U.S. alone with this disease.
Wow.
You're correct. The registry itself is closing in on 2,000 or so people globally.
That's worldwide, right?
Worldwide, but one thing I'll say, it's really important. Registries are massive lagging indicators of the true size of a disease. And there's many reasons for that. But, for example, we've looked at other analogous diseases, spinal muscle atrophy. There was only 3,000 patients in that registry before the drug went in the clinic, and now there's close to 15,000 patients. The drug's approved, and there's many more patients than that diagnosed. So this disease, as you know, Syngap was only recently only in 2021 got an ICD-10 code, and
right.
So I think we're on our way to identify many more patients, and I think there's a lot of really good things happening in terms of also changing diagnostic journey from 10 years down to pretty quickly, right, through whole genome sequencing and other technologies.
Right. How expensive is it to carry this on? I know that you all, when you announced this regulatory approval in Australia, it opened up another $50 million tranche of private placement that went in. And by the way, for anybody who's following along at home, Camp4 is publicly traded, small market cap, but it is publicly traded on the market. What does this mean at that point? And because it just costs so much money to run these trials and to get to the point where you can manufacture a drug.
You're not wrong at all. We have the capital to fund the study and flip the card over and ask the question of whether or not this drug can help patients with Syngap1. And so, what we've guided people publicly is and we have this coming out next week, is our run rate will take us past the study completion, the phase 1/2, right? Mm-hmm. So certainly not enough money, Becky, to bring it to approval yet, but enough money to do the most important test and essentially show this drug is safe and can potentially have an effect on the Syngap1 patient community.
What kind of effect are you measuring for? Obviously, the safety, toxicity, those are key questions. Can the body tolerate it? But what do you hope can happen? Because there have been questions for a long time that if you add a protein back, whether you can actually see the benefit from that or not, especially the older you get. Is there a time limit when it closes, when you still have that synaptic elasticity?
That's such a good question and an important one. I mean, I'll try to be brief with this, but look, the best possible outcome is a cure. That's what I know you have and other parents have in mind, and we should all hold that bar really high. Our treatment approach is meant to treat the underlying cause of Syngap1, as we've talked about, and put more protein, healthy protein, back in the system. We don't know exactly what to expect yet and when, because nobody's ever done this before, but based on the work we've done preclinically, based on the work, for example, there was a recent paper that came out that showed later in life, you can actually, if you put the protein back in, synaptic transmission and connections come back together. So to me, that gives hope that you might be able to have an effect later in life in this disease as well, which is important. ultimately, as you know well, this disease affects so many domains of a patient's life, right? Between seizures, sleep, motor, cognition, socialization. We would like to believe our drug and putting it back in can have an effect across all these different domains. And so what we're doing in this study is what we're calling signal seeking. We've told people, look, we're not going to set a definition for success because we're going to let the data guide us, but we're going to measure everything because we think there can be an effect there, and we want to give these patients the best opportunity to show that there can be an effect there. And so we're entering this study with safety in mind, but using every technology available to us to essentially measure any type of response that can happen from this drug in the patient community.
How do you separate it from, parents who are desperate to see some sort of progress and want to believe in this? Is there a double-blind placebo in this? How do you measure it? Or is it measured against the natural history studies that you mentioned earlier?
We think that it's an important question. We think controls are important. And there's a balancing act here because we also want to do something ethically correct for these patients that are agreeing to enter the study.
Right. The idea of giving your child a spinal tap essentially for a 50% chance at a placebo, I don't think I'd sign my daughter up for that.
I can't imagine how that decision would be taken. So the way we're approaching it is, and let me just speak for a moment about this because it's important. So the reason you want to control, obviously, is to really prove without a shadow of a doubt your drug works, but also because different regulatory agencies have different bars in terms of people ask about accelerated approval or being able to move forward quickly. Ultimately, the goal here is not just to show the drug works, it's to get it on the market to help patients, right? That is the goal we all want, and to do it as quickly as possible. And so sometimes we do things that might seem like, hey, couldn't we move more quickly on the front end? Because we think it offers us the opportunity to get to the promised land more quickly. And this is an example of that. So our intention is to have a study with a control. However, the study design allows for patients that do in fact get the control to be crossed over and get drug as well, so that we're not asking patients to only get a control and nothing else. So
meaning that if it shows up that this is working in the patients who are getting the therapy, you don't have to stay on that control. You can get into the
That's completely
group that's
correct.
Getting the drug.
That's completely correct. And so we have what's called an open label. And so patients will be able to continue to get drug, and those that were in the study would also be able to roll on and continue to get drug at the dose that we identified. And so, because this is a first phase 1/2, we're studying multiple doses, so we have to identify that dose. But we're being very mindful of making sure that any patient that doesn't get drug immediately that's enrolled has the opportunity to eventually get on drug as part of that study. You are correct. There is a natural history study we'll leverage, and we will have a run-in period where each patient can be their own control. So we're doing this because if we get those results we talked about, we want to be able to have the most optionality or robust discussions with the regulators about how quickly can we move this forward and get this to where it needs to be, if there's a place for that.
What does success look like for you? I realize you don't have maybe the primary or secondary endpoint. You're going to be measuring all of these things, but what will make you say, I don't know, a year from now, hey, this worked?
It's a complicated question, and I want to answer it first from, hopefully, if it comes right with a patient perspective. So we have gotten very close to the Syngap community. We are really collaborative with them. I mean, it's, we have a high bar for them. And, from speaking with parents and listening to their stories, success can be from one parent lowering seizures, from another just hearing your daughter or son speak, to allowing your guard to go down for a moment so your child doesn't elope into danger. It's different for everybody, but everybody knows these are all the different aspects of the disease that affect them. So from where I sit, being able to help any of those domains as a starting point is meaningful. And it's meaningful because it can help a family, but more so it can allow the science to be furthered and for other types of technologies to come in. That's how our industry works. I'd love to say our drug would immediately be curative, but the reality is this is a very hard disease. We have a high bar, and we want to be successful. We want to create success for the community first and foremost. Ultimately, for us, if we arrive out of this study with a safe drug, knowing what the dose is that can help patients, and being able to point to different domains of the disease that have moved in a direction where you wouldn't expect that but for the drug, I think that's a starting point for success because it creates a path to move forward and essentially start to help patients and essentially get them to the next level towards being healthier.
Yeah, I hear you on all those levels. My daughter falls in the category of nonverbal, mostly nonverbal, and if she could tell me where it hurt or how it felt, that would be huge. what do you need to see happen? And again, if you have success in this, what diseases would you target next? How would you go about that? I assume you guys have a short list of candidates that you're already focused on.
We do. There is an entire category of genetic haploinsufficient central nervous system diseases that are similar to Syngap. And, interestingly, if you go to the Syngap conference, Cure Syngap conference, which is wonderful, and the families are there, interestingly, related developmental epileptic encephalopathies, which have essentially the same situation that we've talked about here, are right next door. So there's incredible collaboration happening here. And for us, these are exactly the situations where we believe our technology could be helpful as well. So intrathecal delivery, same regions of the brain, extremely high unmet need, and opportunities where if you could put protein back in the system, hopefully we can have a really meaningful effect. So we are having an analyst day in September 28th. I'll put a plug in for it. We're going to talk about our study design. We're also going to talk about 2 or 3 other diseases that we're going to be working on, or are working on, I should say, that are going to follow Syngap. But, look, we've got to get this right for the patients, for the company.
Have you dosed the first patient yet?
We have not yet. Next steps there are to get the sites up and running. And first and foremost, Australia and Argentina. We are continuing to put other regulatory jurisdictions online. We mentioned the US, but also Europe. And we plan to initiate the study in Q4 of this year, which means we'll be treating patients throughout 2027 as well. And we'll continue to give guidance because I know people are very eager, as are we, in terms of learning about when data will become available.
Okay. Josh, I just want to say thank you again for what you're doing, because it just gives hope to so many, not just in the Syngap community, but in the rare disease community writ large. It's an area that anybody living in it feels like it doesn't get enough attention. And to see somebody who's really digging away at this. By the way, I should say, you mentioned that you had worked in operating roles at Biogen. You were in rare disease business development at Genzyme. You've got a biology degree from Washington University in St. Louis and your MBA from the University of Michigan. So you're coming at this from all the important angles. You understand the business aspects, you understand the science aspects. And I just want to say thank you for the work that you're doing.
Look, it's a privilege we take seriously. I'm speaking on behalf of the company, and thank you for giving us the opportunity to talk to you about this, and we won't let you guys down.
Okay. Give us updates when you can.
We will.
Josh Mendel-Brehm, again, is the CEO of Camp4 Therapeutics. And thanks for your time today, Josh.
Thank you, Becky.